All right, so today our topic is going to be short and it should be on molecular motor proteins. We're just going to talk about movement within the cell. So I'll just start the lecture by having you guys watch a 3-minute video.
Cells are full of specialized components that perform functions vital to their existence. But how do these components get to the right locations inside the cell to perform their functions? For larger components, a transportation system is needed. Meet the Kinesin. Masterpieces of microengineering, Kinesins are miniature motorized machines that carry cargo from one part of the cell to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, Kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations called binding sites that interact with other molecules. One site attaches to the microtubule and the other binds with ATP, the energy molecule of the cell. When one foot binds with ATP and uses its energy, the foot flips over, resulting in the walking motion. Each foot has a short neck, which is connected to a strand of a long coiled stalk. At the end of the stalk is a fan-shaped tail, which holds tightly to the cargo being transported. Kinesins can carry cargo that are many times their own size. Sometimes a kinesin is in danger of getting stuck on the microtubule highway because of blockages caused by other cellular components. To get around such obstacles, multiple motor proteins may be used to carry a single piece of cargo, together providing enough force to break free. Kinesins typically walk away from the center of the cell and toward the cell's periphery. The kinesin's 2 feet work together efficiently, with 1 foot holding fast to the microtubule, while the other releases itself and takes a step forward. This coordinated stepwise movement allows kinesin motors to walk as many as 100 steps per second, moving about 8 nanometers with each step. When not carrying cargo, Kinesins can shift to energy-saving mode to conserve fuel until their next job. The kinesin plays a vital role in many cellular processes, not just transporting materials, but also aiding cell replication. The walking kinesin molecular machine, another example of intelligent design.
So what did you learn from that? You should learn the entire lecture. I know I don't plan to spend the whole time with you today.
Yes, we have the kinesins that have on one end 2 feet that can move along the microtubules within the cell, form the skeleton, so it allows them to move in locations, move between locations in the cell. And on the opposite end of the kinesin, you have organelle or whatever other component needs to be moved across.
Wow, that's great. So can you make a wild guess as to the type of organelles that these kinesin or dyenin would be transporting?
Like Cytoskeleton,
Yeah.
or
Vesicles.
cules, vesicles, like and bind. So for exercise and endocytosis.
Yep. So that's basically cellular transport in a nutshell. Well, a synopsis somewhat. It's more complex, highly complex, but that's just a way of simplifying it. But it's literally protein structures that are walking on other protein structures within the cell to transport cargoes, basically. So it's like, imagine a road. Imagine a truck carrying goods from one location to the next. That's basically how things are transported from the nucleus to, for example, an axon terminal, or from an axon terminal to the nucleus. You've seen the different directions in which things can be transported. Today we will be talking about molecular motor protein. It's basically what you just watched a while ago. We'll be talking a little bit more on kinesin, microtubule, cell cytoskeleton. Relating this to mechanics, we'll have to talk about cell cytoskeleton. Just like you have your skeleton, organisms have a skeletal structure that helps it to move or helps to create mobility. It's the same way the cells have cytoskeleton that helps it to move or help to facilitate movement within the cell, around the cell, outside of the cell. So there are specific cytoskeletal structure that are called actin. You already know actin is very important. You met actin when we were talking about muscles. You know that actin interact with myosin and it helps with contraction. So that's one way in which the cell creates movement. You have the microtubule, and as you've just seen, the microtubules are protein tubular structures that exist within the cells, where you have cargoes being transported on these microtubules. So you have other proteins such as dynin, kinesins that can attach to vesicles, Golgi complexes, or particles that can transmit things from the negative end of the microtubule or the positive end of the microtubule. You also have intermediate filaments, which are cytoskeletal structures that are also important. Not necessarily so much with movement, but they're also important structures in the cell that also sometimes aid in movement. The cell cytoskeleton is important for structural support, as you guys should be familiar with. They help with movement, transport of substances, as you've seen. They include microfilaments. Microfilaments are actin filaments. So if you look on the diagram here, you see the microfilaments. Microfilament include actin filaments. We're not going to talk about microfilaments because you guys are well familiar with type of microfilament, actin filament, and actin filament interacting with myosin. We also have intermediate filament. Intermediate filament are protein as well, rope-like proteins that help to stabilize the cell. So it gives it more structure and shape. So not necessarily so much movement, but naturally if the cell has to move, then you expect some conformational changes in the intermediate filament. In terms of the microtubule are the most important, where movement of intercellular structures are concerned. So like the organelles moving from one end of the cell to the next, you'll be looking at the microtubules, and microtubules are very important when it comes to that. So actin filament, we already know enough about actin filament. Actin filament in terms of the structure, you know, the proteins that can be attached to it. This is just giving a comparison of the three cytoskeletal structure. The microtubule consists of monomers of tubulin, alpha tubulin, beta tubulin. These come together to give you, eventually gives you a polymer. So it's just a combination of monomers that form polymer, and eventually it forms a tubule. And this is how you have the microtubule. In terms of the microfilaments, as we say, it's the same. You have actin monomers that come together to give you actin filament. And you know, there are also other proteins that are associated with the actin filament. And in terms of the intermediate filament, it more gives structural support to the cell. So in terms of intermediate filament are much more flexible than actin filament, and microtubules, they range in diameter from 8 to 12 nanometer in diameter. And in terms of the comparison, actin filament tend to be small, the smallest, I think. Intermediate filament tend to be a little bit bigger. Microtubule tend to be much bigger than the intermediate filament. So microtubule is around 25 nanometer in diameter. So you have different classes of intermediate filament, and I think you probably met them in some of the lectures, like the pimentin, desmin, keratin. You've probably seen them somehow, somewhere, I'm sure. And you also have the neurofilaments, which you have in the nerve cells. So these are just adding to the structural composition of the cell, or creating the structure and as well as the shape. So if you look at this, there's the keratinocyte. You expect the keratin to be the intermediate filament, giving the structural or making up the structure are giving the shape and form of the keratinocytes. They're more static in nature than the actin filament and the microtubule if you really compare the intermediate filaments. This is just giving an example of desmin being an intermediate filament that is basically closely associated to the Z disc in the sarcomere, which you guys know. So they don't participate in contraction, but they maintain the structural integrity, and there can be mutation in desmin as well that can cause muscle disorder. So in terms of the function of cytoskeletal moto in cells, cytoskeletal moto proteins are important for membrane transport, as you already know. Nucleus is transported in the cell in the embryonic period. There's a lot of migration, a lot of cell movement that's going on. These cytoskeletal proteins are very much crucial in movement, migration, movement of nucleus, movement of organelles within the cell, even outside of the cell. And these are very crucial stages in cell development and even cell division. So without these motor proteins, the cells basically couldn't exist. Even movement of melanosomes. Would anybody guess what melanosomes are? What are the function of melanosomes?
Involved in the formation of melanin.
Melanin, exactly. So they're responsible for pigmentation. So you realize that when you go into the sun, I'm sure it happens to anybody who goes into the sun for extended period of time. What happens is that sometimes these melanosomes can move from one location to the next. Depending on the exposure of sunlight, melanosomes can actually migrate from the periphery of the cell to the internal part of the cell or to a different location. So it basically changes the external appearance. You might look darker or paler depending on the migration of the melanosomes. And the melanosomes are actually moved by these cytoskeletal motor proteins. Even for transport of viruses, some virus can be transmitted from the end of the nerve terminal. They can infect the nerve at the terminal and be transported from the nerve terminal all the way up to the axon into the cell and infect the nerve cell. And in order for these viruses to be transported from the nerve terminal within the cells, more than likely microtubule, these motor proteins that are basically causing all of this to happen. In terms of transport of messenger RNA, all of these activities that take place in the cell, it will require motor protein. So if you look at this diagram, this is showing kinesin, this is showing microtubule right here, this is showing a kinesin, this is showing a Golgi. So you basically see that there is the kinesin here adhering to the microtubule, and at the top you have the Golgi, and it's moving to a specific direction in the cell. So that's basically a mechanism of transport as well. So what are molecular motor proteins? Motor proteins are they're molecular proteins that use ATP hydrolysis to move along cytoskeleton. Everything in the cells, any form of activity that is required, it's usually ATP dependent. Everything requires energy. There are different different types of motor protein, and we'll focus on kinesin, dyenin. Just to give example, in terms of the kinesin, in terms of the structure, the dyenin, if you look at the diagram here, you can see that there's a motor domain on all of them. And this domain, this area is usually attached to the microfilament, and it basically uses this to walk, right? And if you look at here, you see this looks like the endoplasmic reticulum. This is attaching to endoplasmic reticulum. This is attaching to a vacuole. Here, this is a secretory vesicle. So usually the motor domain is on the microtubule that is walking with this big heavy load on its head wherever it's going, towards the negative end of the microtubule or the positive end of the microtubule. It's the negative end of the microtubule is towards the nucleus. The positive end of the microtubule is away from the nucleus. And kinesin usually take things towards the positive end or away from the nucleus. And dyenin usually take things, vesicle, whatever it is, towards the minus end or towards the nucleus. So if you imagine like a nerve terminal, you would have, for example, dyenin walking on a microtubule on the end of the nerve terminal, taking back protein structures. If you think about kinesin in the nerve or in axon or in the soma, the kinesin kinesin would be walking with a neurotransmitter that is synthesized somewhere in the NISC bodies, synthesized from the NISL substance, and basically take that neurotransmitter along the microtubule to the axon terminal, and naturally the neurotransmitter would be needed at the nerve terminal, possibly in vesicles for initiating an action potential. So in terms of the structure, you guys don't really need to know so much about the structure. Just know that you have motor protein, kinesin, dyenin. But this is just to explain a bit more in terms of the motor protein. Kinesin, if you look at the structure, it has like a light chain here, has a light chain, heavy chain, and similarly, the dyenin can have many intermediate chains as well and light chain. But the important thing is that they have a motor domain, and they also have a region, or motor domain is the head region. They also have a region that attaches to the cargo, whatever it needs to carry towards whatever direction. So they can bind either on actin or microtubules, and you know the mechanism of actin in terms of movement within the cell. Motor protein functions for constant motion, to transport goods within the cell, to transport vesicles, protein. They're important in the embryonic stage, as I said, for division, for forming tissues. So without these motor proteins, the cells couldn't form tissues. All tissues requires motor proteins. So if you look at all of these tissues and you never know anything about motor proteins, it wouldn't make sense to learn about all these tissues if you don't know the origin of how these tissues were created to begin with. So this is just showing also another structure of a motor protein. The motor domain, as I said, the head is the motor domain, which is right here, and the motor domain is like, I don't know why they call it head. I guess it walks on the head. It's walking on the microtubule, and usually you have a cargo attached here. I mean, I would say the cargo should attach to the head, but I don't know why it's walking on the head. And then you have a stalk in the middle, and you have the neck linker that links the stalk to the head. Cytoskeletal motor protein, as I said, the head region is the motor domain that binds and hydrolyzes ATP. So each time it makes a step on the microtubule, there has to be ATP involved. So there's ATP hydrolysis each time in order for it to make each step. So through mechanochemical cycle of filament binding, conformational change, filament release, conformational relaxation, filament rebinding, the motor protein and its associated cargo move one step at a time along the filament, and the identity of the track and the direction of movement along it are determined by the motor, the head. So the head determines where it moves, and the identity of the cargo and therefore the biological function of the individual protein is determined by the tail. So the tail of the motor protein basically determines what it carries. This is also showing the three common motor protein, kinesin, myosin, which you already know of, and dyenin. This is showing the motor domain again. This part attaches to the microtubule, as you know, and it has the stalk and associated polypeptide. What's important about them, they differ in direction that they move along. So they differ in terms of the function and the direction that they move. So dyenin will move the opposite direction to kinesin, and they carry membrane enclosed vesicles or organelles like mitochondria, Golgi, stacked secretory vesicles to their appropriate location in the cell. And this image is just showing kinesin, myosin, dyenin. This is the centrasome, so this should be closer towards the nucleus, right? Closer towards the nucleus, the end of the nucleus. And if this is kinesin, this is the microtubule. Kinesin is walking away from the nucleus or the centrasome, while dyenin is walking this direction. So basically it's just showing that kinesin and dyenin walk the opposite direction. So kinesin is moving towards the positive end of the cell or away from the nucleus, while dyenin is moving towards the nucleus or towards the positive end of the cell. And it's also showing dyenin moving on intermediate filament in the same direction. So this is just showing that this is the plus N complex. So the types of motor proteins that I want you guys to know, you already know actin, you already know myosin, dyenin, kinesin, we just never talk about them. They're just important structures within the cells that are moving along, moving stuff on microtubule, vesicles, Golgi, all of these things within the cell, transporting them from one location to the next. And this is just showing the cargo binding domain, tails, so where the vesicles, vacuoles, whatever it is that needs to be transported is usually attached. And this is just showing the head domain, the motor head domain that is usually walking on the microtubule. So the microtubule are polymers of tubulins that form parts of cytoskeleton. They provide structure and shape to the eukaryotic cells, just like the intermediate filament, but they are more so important for transport within the cell. Microtubules can grow as long as 50 micrometer and they are highly dynamic. So they keep reassembling. If you watch the video keenly, you could see them reassembling each time while the kinesin was just walking on it. And as I said, they have beta tubulin, alpha tubulin. They're just polymers of beta and alpha tubulin combined. And we already know that they can transport a wide variety of cargo, including protein, vesicles, mRNA, and organelles. And they are closely associated with kinesin and dyenin. And this is just showing what you already know. The kinesin moving towards the positive end and the dyenin moving towards the negative end. So this is the microtubule. And as you imagine, these are the head domains. And this is the cargo on top of it. And if they're moving away from the negative end, it's anterograde cargo. Remember I said that in axons you can have anterograde transport and retrograde transport, and in terms of the dyenin moving towards the minus end, that would be a retrograde transport. So this is just giving a bit more detail in terms of the microtubule. You already know that they are dimers that form polymers of alpha and beta tubulin, and they assemble into protofilament. Typically 13 of these protofilaments then align to give you the hollow tube. So it's like the beta and alpha tubulin monomers just form in group. These are made in the centromeres. You'll eventually get to understand. This is why I put this little diagram down here. The centromere is an important organelle in the cell, and we never really talk so much about the centromere, but now you know that the centromeres are important for microtubule assemblies. The synthesis and assembly of the microtubule is basically coming from the centromere. So the microtubule have a lot of function. The centromeres even have a lot of function. Microtubules, they are important in cell division process. So when the cells are dividing, they form centromeres on either side and they pull the cell apart. And there's also microtubule that is a very integral structural component in cilia, flagella, and they are dynamic. As we know, they're always changing. They can reassemble or keep assembling alpha and beta tubulin monomers, and they grow from the centromeres. And nothing that we haven't mentioned before, the important thing is that in terms of the movement, this is just showing myosin acting on actin. Yeah, you know that ADP is when it's in the resting state, and ATP is basically for the contraction to take place. And it's the same. Actin, dyenin, kinesin needs ATP to actually walk on the microtubule. So myosin motor power movement on actin filament, you already know that, whereas dyenin and kinesin motor power movement on microtubule. So it's like actin and myosin, dyenin, kinesin on microtubule. And this is just showing the myosin on the actin and the dyenin, kinesin on the microtubule. So the cytoskeletal tracts we would call them the microtubule. So the cytoskeletal tract would include that intermediate filament, but mostly the microtubule that are just there for the dyenin and the kinesin to just transport stuff on. And this is just showing the same thing and the direction in which they're moving. In terms of the cilia and flagella, they have built-in microtubule and dyenin. Just as microfibrils are highly specialized and efficient motility machines built from actin and myosin, cilia and flagella are highly specialized and efficient motility structure built from microtubule and dyenin. So remember that cilia and flagella, or cilium, flagellum, they are built from microtubule and dyenin, and you will get to realize that it's bending. These are the microtubule. The axoneme is basically just microtubules. This is cilia membrane, and if you look at it closely, you can see that there are cargo, kinesin, dyenin that are also associated with the microtubule that is a part of the cilia. It's bending of this, so they call it axoneme, but it's microtubules that are there, right? It's bending of this that creates the movement in the cilia or the flagellum. And this is just showing the arrangement of microtubule in flagellum or cilium. This is microtubule. We expect tubular structures, and it's basically just showing their arrangement. And this is just an electron micrograph showing the arrangement of microtubule in cilium or flagellum. So there are contrasting motion of cilium and flagellum. Yes, it's not so important that you know them. Just know that it's the structural protein that is important for the movement. You're an engineer, you guys are engineers. If you're going to synthesize or create anything, it's likely going to be protein or protein structures to assist in some dysfunction or dysregularity. So it's more important to know how to actually already synthesize the protein itself than the actual mechanism. The mechanism is easy. So this is just showing that flagellum, it moves in like a propeller-like motion, but like the cilium is more like a back and forth beating motion. The axoneme is composed of microtubule and their associated proteins arranged in a distinctive and regular pattern. This characteristic arrangement is found in almost all forms of eukaryotic flagella, cilia, from protozoans to humans. And in terms of the microtubule we said are synthesized, or the organizing center of the microtubule is the centromere. And you know the centromere is an important organelle in the cell, and it's close to the nucleus. It's important for construction of the microtubule. Centriole are two units making up the centrasome. These two units, centriole, centriole, this makes up the centrasome, and they're arranged at right angle to each other, as you can see right here. And centriole basically contain 9 triplets of microtubules. So if you count 1, 2, 3 and count all the way around, it's the microtubule triplet here. And we already mentioned that microtubule plays a very important role in cell division, where you have the formation of the mitotic spindle. So if you look on the diagram, you can just look at this in your spare time. Nothing special to understand now. I mean, it's just microtubule and centromere pulling the cell structures apart. So we never touch on replication, but when we talk about microtubules, then you really have to talk about replication. And if you talk about microtubule, you have to talk about centrosome or centrioles, and then that's where the replication comes in. You guys already probably know cell replication from high school biology, so not going to spend much time on this. But the cell cycle, just like you have a lifespan and a cycle, the cell also has a lifespan, which we call a cycle. Cell might not live as long as you live, but they also have a lifespan and a cycle, right? So in terms of the phase, we divide the cell life into phases or cycle, different type of growth phase when different things happen in the cell. So there is the G phase. Sometimes some cells can go into the dormant stage where they don't have to replicate replicate immediately. Could you give me example of cells that would go into a dormant stage that don't necessarily need to replicate?
Sorry, miss, can you repeat that once more?
Example of cells that can go into a dormant stage. It's not going through a growth cycle. It's in, for example, a G0. Hepatocytes, they replicate, yes, but they can go into a dormant stage. What about permanent cells? Cells that just don't replicate. You look at neuronal cells, no? Yeah. How about skeletal, cardiac, muscle cells? Yeah, they're not replicating. They're not just going to constant turnover. So what are example of cells that are always replicating, or there's constant turnover? Always in a growth phase, always
hematopoietic stem cells.
Yes, very good example, hematopoietic stem cell. How about epithelial cells? Remember the skin? You're constantly shedding cells off of the skin every day. So these cells are constantly recycling. How about the gastrointestinal tract? These cells are constantly turning over. The genitourinary system, these cells are always dividing, right? And they also have cells that will go into dormant stage, but they will replicate. For example, your hepatocytes, right? The hepatocyte cells from different glands, pancreatic cells. These cells can re-enter the growth phase at intervals. But as it relates to cells that are always dividing, the epithelial cells on your skin, the gastrointestinal cells, the enterocytes, they're always changing over. And then you also have permanent cells that are not always replicating. For example, the cardiac myocytes, they tend to not replicate after a period of time, and even the neuronal cells as well. There are different stages in the cell cycle. The G1 or pre-synthesis stage. The G is the growth stage or growth phase. The synthesis stage is basically when the cell is preparing to grow or preparing to replicate. So it has to synthesize proteins, it has to synthesize enzymes, it has to replicate the DNA in order to get into that synthesis stage. Because if it's actually going to replicate, remember that it's going to split in half. So it has to really try to prepare up the organelles, make some organelles, make some protein, divide its genetic material so that it can share with the other cell. So it goes into that synthesis stage. After the synthesis stage, it goes into a pre-mitosis state. So before it actually split in two, or before there is actual mitosis or replication of the nuclei, the nucleus. So the DNA has to replicate, then the nucleus has to replicate, then the cytoplasm has to splitting off, the cell membrane splitting off. Once the genetic materials are synthesized, then it's basically divided. G1 to the synthesis, to the G2, which is the growth phase, to mitosis, cytokinesis, where the cell actually splitting off. And in terms of the mitosis, there are different stages or different stages of mitosis. Anybody remember anything about mitosis?
Yes, we have anaphase, prophase,
Anaphase, prophase, is that the right order?
Why? That's to remember.
All right, at least you know that it exists. So at least you remember your high school biology. You don't have to commit the details to memory, but at least remember the order.
As in the events that occur.
Yeah, the events that occur. You know that there is also mitosis, meiosis. I'm not going to go through the details, but you should know that at least there's the prophase followed by the metaphase, followed by the anaphase, followed by the telophase. And the mitosis is different from meiosis in that at the end you have the division of the daughter cells. You have 2 cells in mitosis versus meiosis, you end up with 4 cells. And you know that mitosis, all the cells in your body divide by mitosis except the gametes or gametocytes, like the sperm cell, the eggs. These are the only ones that basically divide by meiosis. And it's the same. You have the prophase, the metaphase, the anaphase, the telophase. It's just that you have it times 2, because at the end you end up with 4. So you end up with 4 daughter cells in this stage as opposed to 2. So the replication cycle is the same for both of them. It's just that you have 2 phases of prophase, metaphase, anaphase, telophase, 1, and then you have 2. because the cell has to divide twice. In terms of cell motility, in all these cells, we know that a lot of cells are motile. Do you remember any motile cells?
Macrophages.
Macrophages are motile. What fibroblasts are motile?
Yes, miss.
Okay. All right. So yeah, these cells are motile. They're basically just using cytoskeletal structures, and some of them have pseudopods or cilia, flagellum for motility. For example, if it's sperm, it's using the flagellum, and know that the microtubule is an important structure within it for the movement. And some of them actually crawl. They use even the intermediate filament and create conformational changes in certain structural component or even pseudopods. If you remember, like even the osteoclast, osteoblast, all of these cells that are moving around, when you look, you see them have the extent areas of their cytoplasm. We tend to call that pseudopods or pseudopodia, and they actually use that to crawl around. So yeah, a lot of cells are motile for very important processes. For example, wound healing, cancer metastasis. These cells are moving all around. They're not supposed to be moving. Even when you have cancer, and for example, you have a prostate cancer, and sometimes cancer end up into the brain, it end up in the spine. It's the cells that are dividing and are moving and are invading different areas that they were not originally designed to invade. So there are a lot of genetic signaling or dysfunction in these cells that are creating abnormality, right? In terms of immune response, a lot of migration has to take place. Even in bone healing, a lot of migration or inflammatory process has to take place. And we know that the neutrophils, fibroblasts migrate. In terms of a kinesin, kinesins are microtubule-based protein that are well known for their role in biological process. They are important in cell division. So even when you look through the division of the cell, the replication, we have the mitotic spindle. So when you go through the mitosis and meiosis, you will see that there is the mitotic spindle. And there's also kinesin that is important for movement, mobility of these cells. And this is just showing the structure of the kinesin and the fact that ATP is a very crucial part of the activity. And there's a large family of kinesin. So even though I'm saying kinesin is not one type of kinesin, you have a super family, and it's the same with Dyenins. So in science or even in medicine, sometimes we simplify by saying, kinesin. You have different types of kinesin based on the structure. So when you do like a genetic sequence or you dissect the structure itself, you realize that there are components to, for example, this one that is different. So you have to sub-classify or put it into family or subfamily. And it's the same for viruses, bacteria, parasites. You have to categorize them based on distinct differences, the most minute differences that you see in them. So you have a large family of kinesin 1, 2, they can be hetero, trimeric, homo, dimeric, depending on the kinesin chains, light chains, or different structure or structural composition of proteins that are associated with it. We know that kinesin are a large superfamily of proteins that are responsible not only for trafficking cellular cargoes along microtubule, but also for controlling microtubule growth. They also help with microtubule growth, and there are a lot of kinesin genes, there are a lot of Dyenin genes, and even actin, which is why you have different types. And this is just showing different kinesin. If, for example, you do sequence, you have different sequences, you give them a session or subcategory based on a specific feature. So this is what these would be in that case. Different types of kinesin. And in terms of the development of the central nervous system, the kinesin is very important. The kinesin is very important for axonal dendritic process transport and even for neurotransmitter, as I mentioned. You guys can look at this also in your spare time. This is just showing the microtubule in the axon, coming from the axon terminal going up to the cell body. And this is just showing different kinesin walking, kinesin should be walking towards the positive end, dyenin along here should be walking towards the negative end. And even for fertility, all of these proteins are very crucial. So without these proteins, we would not be here. So at least now you know that cells, the origin of cells, the emergence of cells, this division of cells, migration of cells, movement of cells are a result of these molecular motor proteins. And that's the end. Any questions for me?
Yes, miss. What did you say was the positive end and then what is the negative end when you talk about movement?
What I said, what it was?
Yes, so the positive end refers to...
So for simplifying it, use the nucleus as your start or your base point, or closer to the centrosome. The nucleus would be, if you're moving from the end of the cell towards the nucleus, that would be the negative end. Or away from the nucleus towards the periphery of the cell would be the positive end. So think about like the axon, think about the neuronal cell with the cell body, the axon, the cell body moving stuff away from the cell body towards the axon terminal would be moving it towards the positive end, and moving it from the axon terminal towards the nucleus or the cell body would be the negative end. So you would have dyenin moving stuff towards the negative end, which would be towards the nucleus, and kinesin moving cargoes towards the positive end, which would be away from the nucleus or the centrosome. And either way, once it gets to the end of the cell, there are different mechanisms of release, and that's when the mechanism of release comes into play. The exocytosis, transcytosis, pinocytosis, all of that we actually covered in the first lecture. Any other question? You still there, Daniel?
Yes, I'm still here. No questions.
You surprised me done so quick, no?
All right, thank you, miss.
Yeah. If you have any issues, problems, concerns, I'm always reachable, as you know.
So like the final exam itself is going to be of a similar structure to what we got yesterday?
Similar structure to yesterday, but you'll get 50 questions for 2 hours.
Okay.
1 mark each. So not like the 0.5 that you do in the tutorial. I think you guys do a lot of work for the tutorial. That's where most of your assessment was weighted for the tutorial. So the final exam should just be 50 questions for 2 hours, but it's short answer. Just whatever I ask you. You don't have to write sentences. I really don't care. It's not an English class, and I don't check for spelling. I understand that it's science. You don't need to learn to spell as long as you know how to save lives or create a molecule. Nobody cares if you know how to spell it. As long as I can understand somewhat, that's what it is. All right, so you guys just let me know when you're ready for the revision session, and good luck on your endeavors or whatever else you need to do.
It's appreciated. Thank you so much for helping us out this semester.
No
Learned
problem.
a lot, and I must say I enjoyed the course.
I'm happy you did. That was the idea, and I'm happy that you did. I hope you all actually did enjoy the course.
Yes, miss. Thank you. Same to you.
Yeah, thanks. Bye.